Parkinsonism is a clinical syndrome that covers several conditions, including Parkinson's disease and other conditions with similar symptoms, including slowness of movement, increased tone in form of rigidity, tremor, and problem with walking. 80% of Parkinsonism is caused by Parkinson's disease. The rest of causes including cerebrovascular disease, drug and toxin, genetic, anoxic brain injury, and other neurodegenerative disease. A small number of people will develop Parkinsonism after taking certain medication. Those generally block the action of dopamine, including antipsychotic drug, metoclopramide, prochlorperazine, tetrabenazine, sodium valproate, lithium, and manganese. The symptoms of drug-induced Parkinsonism is not progressive, and often most people will recover within hours or days of stopping the medication. Also, people with Parkinsonism may have their symptoms get worse after using the same medication. Other causes of Parkinsonism is vascular or multi-infarct Parkinsonism, which is a form of Parkinsonism that produced by one or more than one small strokes. Patients with vascular Parkinsonism might have the same symptoms as idiopathic Parkinson's disease, but often it's involved the lower part of the body rather than the upper part, with association with focal neurological deficit, often asymmetric limb weakness, numbness, abnormal reflexes, or abnormal speech. The CT scan or MRI of brain is usually abnormal in 90% of patients, which is often showing multiple small strokes in the deep portion of the brain.
Neurodegenerative brain diseases, such as progressive supranuclear palsy or PSP, are debilitating diseases that can drastically impact the lives of patients and their families. Neurodegenerative diseases are characterized by the progressive loss of neurons and are associated with a range of symptoms, including increasing loss of cognitive and motor functions. A fundamental mechanism of neurodegenerative brain diseases is the misfolding of proteins such as tau. Tau is a cytoplasmic microtubule-associated protein that is widely distributed in neurons. Tau contains microtubule binding domains, enabling it to support assembly and stabilization of microtubules, a key component of the cytoskeleton, which are fundamental for nerve cell growth and maintenance. Under normal conditions, tau undergoes several post-translational modifications, including phosphorylation, which regulate microtubule binding. Under pathological conditions, tau becomes hyperphosphorylated, leading to dissociation from microtubules. This dissociation leads to microtubule destabilization. Hyperphosphorylated tau is further modified by processes such as truncation. This pathological tau begins to aggregate by folding into a more compact state and joining together with other tau molecules at the microtubule binding domains to form soluble oligomers. These pathological oligomers do not necessarily remain confined to the neuron in which they originated. They may spread from neuron to neuron by crossing the synapse into neighboring cells, where they are able to stimulate the aggregation of natively folded tau, spreading the pathology. These pathological tau oligomers can also form long insoluble filaments, which accumulate within the cell, leading to cell death and resultant brain atrophy and the formation of neurofibrillary tangles, or NFTs. These NFTs are a key indicator of diseases such as PSP. PSP is a progressive tauopathy associated with a wide range of motor and cognitive symptoms that worsen with disease progression. Symptoms include balance problems, lack of coordination, slowed movement, difficulty controlling eye movements, blurred or double vision, loss of speech and difficulty swallowing, forms of dementia, personality changes, and impaired thinking. The most common symptoms of PSP include gaze palsy and postural instability, which often leads to backwards falls. Eventually, patients may lose almost all motor function and become bedridden. There are currently no approved therapies for slowing or preventing disease progression.
Multiple system atrophy is characterized by symmetric onset Parkinsonism, early and significant autonomic failure, mainly postural hypotension, and cerebellar symptoms. It is much less common than Parkinson's disease, with a prevalence of about 4 per 100,000. The pathological hallmark is alpha-synuclein containing glial cytoplasmic inclusion found in the basal ganglia, cerebellum, and motor cortex. The early distinction between Parkinson's disease and multiple system atrophy may be difficult. The clue for diagnosis is early fail and postural instability with lack of response to levodopa. The management is symptomatic. Prognosis is less good than Parkinson's disease and leading to early disability, with mean survival from the onset is fewer than 10 years. Corticobasal ganglionic degeneration is less common than previous two diseases, and the clinical manifestations are variable, including Parkinsonism, dystonia, myoclonus, and alien limb phenomena, whereby usually upper limb move about or interfere with the other limb without apparent conscious control, as in such patient.
Huntington's disease is a genetic, neurodegenerative, and ultimately fatal disease with devastating impact on individuals and entire families. Huntington's causes problems with a person's ability to think, behave, and move. Over time, the disease causes progressive functional decline, leading to a loss of independence and the ability to perform daily activities. Huntington's typically strikes a person in the prime of their life, between the ages of 30 and 50, although symptoms may appear earlier or later. Today, clinical diagnosis is made when movement problems are obvious. However, subtle changes known as prodromal or premotor onset symptoms often appear years earlier. Each child of a parent with Huntington's has a 50% chance of inheriting the disease, which affects men and women equally. We call this pattern of inheritance autosomal dominant. About 1 in 10,000 people live with Huntington's today, with many more at risk, including over 30 individuals within my own family. Everyone has 2 copies of the Huntington gene, which contains a section of repeated genetic letters known as CAG repeats. The Huntington protein is produced throughout the body, and although the normal function is not yet fully understood, it is thought to be involved in many biological functions, including the development of the nervous system. Huntington's disease occurs when 1 copy of the Huntington gene has a CAG repeat expansion over a certain threshold. People with 40 or more CAG repeats will develop Huntington's in their lifetime. People with 36 to 39 repeats may or may not develop the disease within an average lifespan. And those with 27 to 35 CAGs will not develop Huntington's themselves, but risk passing on the disease to the next generation. The Huntington gene carries instructions to make the Huntington protein. The DNA is used to create a messenger RNA that carries the code to the cellular factory that makes the protein. Too many CAG repeats result in a toxic form of the protein being produced, known as mutant Huntington protein. Mutant Huntington protein damages the normal functioning of neurons in many ways, including interfering with their transport networks and gene regulation. This causes them to malfunction and die prematurely, resulting in the symptoms of Huntington's. Huntington's is increasingly recognized as a disease of the whole brain and body. Areas of the brain responsible for cognition, personality, and movement are particularly affected, leading to the classic triad of symptoms in Huntington's disease.
Huntington disease presents with progressive behavioral disturbances, abnormal movement usually in form of chorea, as shown in these videos, with cognitive impairment leading to dementia. Onset under the age of 18 years is rare, but patients may then present with Parkinsonism rather than chorea, which is named as Westphal variant. The diagnosis is made by genetic testing. Brain imaging may show caudate atrophy, but it is not a reliable test. The chorea may respond to neuroleptic such as risperidone or tetrabenazine. Depression and anxiety are common and may be helped by medication. For understanding Wilson disease, we must know the copper metabolism.
Essential mineral that our body needs to get through the diet is copper, and typically we take in about 1 to 2 milligrams per day from the food we eat, things like whole grains, beans, nuts, and potatoes. But really, our body only needs about 0.75 milligrams per day, so that extra copper is excreted. About 90% of the excess copper is excreted into the bile, where it eventually ends up as fecal copper, and the other 10% is excreted in the urine. In Wilson disease, there's a genetic defect that results in the excess copper being kept in the body and deposited in various tissues, where it's not supposed to be. And just like iron, free copper reacts with hydrogen peroxide in the body to form the hydroxyl radical, a reactive oxygen species that's pretty good at damaging tissue. So over time, those tissues are seriously damaged by free radical generation. Now, your liver cells, or hepatocytes, play a really important role in helping the body get rid of excess copper. So usually, the copper from the diet is absorbed in the small intestine via enterocytes and passed off into the portal vein to the liver. Once it's in the liver, it's sent to a special transport protein called ATP7B, which has a couple super important jobs. The first job is that it binds copper to apoceruloplasmin, which is the major copper-carrying protein in the blood and is responsible for carrying 95% of the copper in the blood. After it binds copper, it's then just called ceruloplasmin, and this guy can haul 6 molecules of copper at once. ATP7B's other job is to gather up the rest of the copper into vesicles to be exocytosed into the bile canaliculi, where it goes into the bile and is eventually excreted. With Wilson disease, there's an autosomal recessive defect in this ATP7B transport protein. As you could probably guess, that means it can't incorporate the copper into ceruloplasmin or excrete it into the bile. Since it's not doing either of these things anymore, the copper builds up inside the hepatocyte and starts to produce free radicals. Eventually, all this built-up copper and free radical damage injures or destroys the hepatocyte, causing free copper to spill out into the interstitial space, and from there into the blood supply, where it's circulated to and deposited in other tissues, where it also causes free radical damage over time. One organ in particular is the brain, and for this reason, Wilson disease can have serious neurological symptoms and complications. Depending on where it deposits, it can cause different disorders. If it deposits in the basal ganglia, it can cause a movement disorder that's a lot like Parkinsonism. If it gets to the cerebral cortex, it can be toxic to neurons and can lead to neuronal cell death and dementia. One place that it can deposit that can be helpful for diagnosis is in Descemet's membrane of the cornea, which is the membrane between the stroma and the endothelial layer of the cornea. So you'll look for something called Kaiser-Fleischer rings, which are visible copper deposits in the cornea.
Wilson's disease is the excessive accumulation of copper in the body and the tissues. It's caused by a mutation in the Wilson's disease protein, which is on chromosome 13, and the Wilson's disease protein also has the catchy name of ATP7B copper binding protein, and is responsible for various functions, including the removal of excessive copper in the liver. Genetic inheritance of Wilson's disease is autosomal recessive. So what are the features? Most people with Wilson's disease present with one or more of these three different systems. Hepatic problems or liver problems, which occur in 40% of people. Neurological problems or problems with the brain, which occur in 50% of people. Or psychiatric problems, which occur in 10% of people. Now, copper deposition in the liver leads to chronic hepatitis, so inflammation of those liver cells that have excessive copper in them, and eventually this leads to liver cirrhosis. Copper deposition in the central nervous system leads to neurological and psychiatric problems. Now, the neurological symptoms can be subtle and range from concentration or coordination difficulties to dysarthria, which is speech difficulties, and dystonia, which is abnormal muscle tone. Copper deposits in the basal ganglia in the brain leads to something called Parkinsonism, and this is where you get three things: tremor, bradykinesia, or small movements, and rigidity. So tremor, bradykinesia, and rigidity. These Parkinsonism symptoms are symmetrical, which is what differentiates them from the asymmetrical symptoms that you find in Parkinson's disease. Psychiatric symptoms can vary from mild depression to full-blown psychosis, and the underlying cause of Wilson's disease is often missed, and treatment is delayed. Kaiser-Fleischer rings are found in the cornea of the eye, and what they are deposits in Descemet's corneal membrane. So this is small circular brown rings that present around the eye in patients with Wilson's disease. And these brownish circles surrounding the iris can usually be seen by the naked eye, but proper assessment requires a slit lamp examination. There's a few other features. Often you get a hemolytic anemia where the red blood cells are being broken down too quickly. You can get renal tubular damage, which leads to renal tubular acidosis, and you can get osteopenia, which is a loss of the bone mineral density. So if you suspect someone has Wilson's disease, how do you actually diagnose it? Well, the initial investigation of choice to screen for somebody who might have Wilson's disease is to do a serum ceruloplasmin. And this is a protein that normally carries copper in the blood. It can be falsely normal or elevated in cancer or inflammatory conditions. So if somebody has cancer or an inflammatory condition and you suspect Wilson's disease, don't rely completely on this result. So it's not specific to Wilson's disease. Some other conditions can cause raised ceruloplasmin. You can do a liver biopsy, which is to test the liver for its copper content, and this is the definitive gold standard test for a diagnosis. So if you want to establish a definitive diagnosis, liver biopsy is the way to go. You can also make a diagnosis based on the 24-hour urine copper assay. So this is how much copper is in the urine over a 24-hour period. But this is quite a cumbersome test where you have to continuously collect urine samples for a full 24 hours. Alternatively, there are scoring systems that take into account various features and laboratory test results to establish a diagnosis of Wilson's. There's a few other investigations that will point you in the direction of Wilson's disease. One is to check the serum copper, and this is usually low in Wilson's disease. To look for Kaiser-Fleischer rings, and to do an MRI brain, which can show, again, some non-specific changes that will indicate Wilson's disease.
Treatment for Wilson disease involves an initial acute decoppering therapy followed by maintenance therapy to keep copper levels within the normal range. Treatment options include copper chelators such as trientine, penicillamine, an alpha amino acid metabolite of penicillin without antibiotic properties, and tetrathiomolybdate, and zinc salts. Copper chelating agents bind free copper in the blood and tissues to facilitate excretion. Also though, patients can be given zinc salts, which increases the production of metallothionein proteins in intestinal cells. This protein binds to copper and traps it in the cell, which is shed and excreted in the feces. Finally, if the liver's been damaged to the point of cirrhosis and liver failure, a liver transplant may be needed.
In summary, features that doubting the diagnosis of idiopathic Parkinson's disease and reconsidering the diagnosis of it are: first, poor response to levodopa, symmetrical feature at onset, infrequent or atypical tremor, prominent rigidity in axial musculature, early postural instability, supranuclear gaze palsy, early autonomic failure, cerebellar involvement, alien limb syndrome, focal neurological deficit, apraxia, and significant early cognitive dysfunction.